All right, so here in 17.2, we are going to be talking about our chemical defenses. So remember, we're talking about our innate, non-specific immune responses, meaning things that we are born with that are innate to our bodies. And then this included what we already spoke about in chapter 17.1, which are our barriers. So our actual physical barriers to allowing a pathogen or the way that we prevent a pathogen from getting into the body. Now what we're going to talk about are our chemical defenses. So what we have in our body, we have some chemical mediators, and these are substances that are found in various body fluids and tissues, so floating around and within the tissues themselves, that can work alone or with each other to inhibit microbial invaders. So we actually create these chemical mediators, to try to prevent pathogenicity, or in order to prevent invasion and infection in our bodies. So there are some different types of chemical mediators, some terms that you should be familiar with. The first one is endogenous, and this means that it's made by the human body's cells. So our cells specifically, these are the ones that our body cells are making, these chemical mediators. These are endogenous. Then there's also exogenous. So exo meaning outside of, exogenous, endo meaning inside. So exogenous are made by certain microbes in the microbiome. So again, we have all of these organisms living in us and on us, and they're helpful to us. These are a microbiome. And not only are they helpful in the ways that we discussed as far as physically preventing things via barriers, where we said that there's competition with our microbiome, and if there's competition, then hopefully pathogens don't get a foothold and things like that. But then also, we make these, or we have these bacteria, we have other organisms in our microbiome that are going to be making these things to prevent pathogens. So not only because of the competition, but because they make exogenous chemical mediators. And so some of these are continuously produced. This is supposed to be bathing here. So continuously produced, bathing the area in antimicrobial substances. So we can constantly be producing these things and just basically making it so that pathogens don't get a foothold, so that they're just constantly having these antimicrobial substances there to try to prevent them kind of at all costs. Then some are produced or activated, or not at all costs, but at any time is my point there. Then some are produced or activated in response to a stimulus. So if then we have this microbe that's present, then what we'll do is we will start to make some endogenous or exogenous from the microbes to try to kill or decrease the growth of any pathogen that might get in the body. So we have endogenous with our human body cells producing these substances. Exogenous are the microbes in our microbiome that are producing the substances. Some of these are constantly being produced to try to prevent any pathogens, regardless of the time. And some of them are produced or activated in response to a pathogen being present or some other stimulus. So it could be the toxin of a microbe, for example. So let's talk about some of these chemical defenses. So first of all, our body fluids. And if we go back to the skin, kind of our first line of defense, we already spoke about it as an actual physical barrier with many layers of cells to prevent any pathogen from getting through. We also have various other body fluids that are associated with the skin. So we have our sebaceous glands in the dermis that are going to produce oil, which is called sebum, and this is released onto the skin surface. So here with the sebum that's released onto the skin surface, this is an endogenous mediator, right? So remember, endo meaning inside of our own cells. So our own cells, our glands, are going to be making this sebum, and then they're going to be pushing that to the surface of the skin. So not only is it going to help to keep the hair moist because it's released onto the hair follicle, but it also then is going to get onto the surface of the skin. And it allows for prevention of bacteria from invading, particularly from invading those sweat glands that are also in the area that have an opening to the surface of the skin. But then also it's going to create this thin layer of sebum or oil on the surface of the skin. So you can see the sebaceous gland here. On the left is an illustration. On the right is actual histology, a slide of the sebaceous gland right next to. This is the hair follicle here. So this gland here is going to make the sebum, and then that sebum is going to go on the shaft of the hair here and then go out onto the surface of the skin. And so this is made by those glands, and then it goes onto the surface of the skin. And this does many different things. So it helps maintain moisture inside of the body, but it also is, and it maintains part of the skin, moisture in the skin, so it doesn't get dried out too quickly. But then it also prevents bacteria from invading, so it gives another layer there to prevent it from getting into the skin and getting, deeper into the body. Also, the way that this works is that our skin microbiome, organisms like P. acnes and Malassezia, can use lipases, though, to degrade the sebum and use it as a food source. So in our microbiome, remember, this is part of the organisms that live on us. We have this P. acnes, which is a bacteria species, and Malassezia, which is a fungus, and these live on our skin. When our sebaceous glands are producing sebum and that goes out onto the surface of the skin, then these bacteria and fungus species are going to utilize their lipases that they make. Remember, lipases are lipids, enzymes that break down lipids. They can then degrade that sebum, that oil, which is a lipid, and then they use it as a food source. So this is one way that we're feeding our microbiome, the ones that are on our skin anyway. When they eat this, when they degrade these lipases and use it as a food source, what it does is it produces oleic acid. When it produces oleic acid, as an acid, it is mildly acidic. And so then what that does is it makes our skin surface mildly acidic. So if we have all this bacteria and fungus on the surface of our skin and it's producing acid, oleic acid, and it's going to make the surface of the skin mildly acidic. And if it's mildly acidic, then that ends up being inhospitable to other microbes. So in this case, this would be an exogenous mediator. So not only do we produce sebum, which is an endogenous mediator that has some antimicrobial properties, but then the microbiome that we maintain on the surface of our skin eats this sebum, and then they produce oleic acid, which then is an exogenous mediator because it makes the surface of the skin a little bit more acidic, and that's not good for pathogens. They don't live well in more acidic environments. So we do have environmental factors that can then affect the microbiota of the skin and then therefore affect the chemical mediators. And we have spoken about this previously where if we wash our hands a lot, for example, it leaves us susceptible to infection because we're, within reason, right? So if we're washing our hands or washing our bodies, maybe a better example. If we're washing our body, very often, what this does is it washes away that microbiome over and over and over again, and so then we don't have this oleic acid building up on the surface of the skin. We also don't have the sebum, therefore we don't have the sebum, therefore we don't have the oleic acid, and so then we are missing out on both of those endogenous and exogenous mediators that help prevent pathogens from invading the body. Also, things like low humidity is going to affect sebum production, or decreased sebum production itself is a type of environmental factor that's going to affect the microbiota. So if we have low humidity, it might kill off some of our microbiome on the surface of our skin. If we are not drinking enough, right, then maybe we're not healthy enough, we're not going to be producing as much sweat, which is different, but then also it's not going to, or it's going to affect the way that our sebaceous glands are going to work. And then if we are decreasing our sebum production, then what we're doing is we're making the skin less habitable for our microbiome. So our actual bacteria are going to die if they don't have anything to eat. So like this P. acnes and the Malassezia are then not going to be able to live on the surface of our skin. If they're not living on the surface of our skin, then that increases the pH rather than being acidic, and then that's going to make us more susceptible to pathogens because they like a more neutral environment rather than an acidic environment. So if we have low humidity, if we're not drinking enough, if we are washing our skin, our body too much, then that can really make it so that we're not producing the sebum and therefore we don't have this protective layer. Another example of a body fluid, a chemical defense with body fluids, is in the digestive tract. So if we start off in the oral cavity of the digestive tract, first of all, we interact with saliva. So saliva is going to produce lactoperoxidase enzymes and then also lysozyme. So our lactoperoxidase enzymes and lysozyme are going to work to cleave peptidoglycan. So remember, peptidoglycan is what we find in the cell walls of bacteria. So our lactoperoxidase and our lysozyme right in the oral cavity. So as soon as we eat something, if there happens to be bacteria in there, particularly pathogenic bacteria, then we have these things in the saliva, these endogenous mediators in the saliva that are going to start cleaving peptidoglycan. So it'll start cutting up their cell wall, trying to destroy those bacteria or break those bacteria down. So then it'll eventually kill the microbes, especially as it's taking time. The more we're chewing in our mouth, we're interacting with the food or mixing the food and the saliva. And if there's bacteria in there, then that's longer that saliva with the lactoperoxidase and the lysosome are going to have contact with the bacteria. And then as we swallow and it gets into the stomach, that would continue. Then once we get into the stomach, then we have our highly acidic gastric fluid that will kill microbes. So again, it's highly acidic in there, just like the acidic surface of our skin, but this is even more acidic. So even more acidic, then it likely will kill those microbes. But if the microbes make it past the stomach, then we can move into our intestines. When we move into the intestines, we have the pancreas that's going to be secreting fluids into the intestines up at that very first portion in the duodenal area. And then we have other intestinal enzymes and antibacterial peptides that are going to be secreted into that first area there. So we have our pancreatic enzymes that are going to be put into the duodenum. So that includes different peptides, which peptides are those that are going to be breaking apart proteins. And so when we're talking about bacteria, they also have proteins on them, within their cell walls, but then also in their plasma membranes. So those peptides or peptidases are going to be able to work on bacteria as long as it's working on our food as well, and various other pancreatic enzymes that are going to be released there that can affect the bacteria themselves, as well as our other intestinal enzymes. So on our brush border, as we get further down toward the end of the intestines, toward the ileum region, which is the third portion of our small intestines, is where we find things like our enzymes that are going to break apart things into individual molecules. And that includes things like lipases to break apart lipids. It also includes various things that break down saccharides, so things like maltase and things like lactase and sucrase and things like that are going to be breaking these things apart. So along the way through our intestines, and we have these various enzymes that are going to be breaking apart little pieces. And the reason that they're there, of course, is for the food, but it also works perfectly to break down bacteria that have these things within their cell walls and their plasma membranes as well. We also have our antibacterial peptides, I was mentioning, called cryptins. And so again, these are going to be specific or more specific to breaking down bacterial peptides that are going to be released. Also, in that first portion of the small intestine, in the duodenum, we have bile that's coming from the liver. And bile is something that sequesters or separates lipids. And so bile is going to assist in breaking things apart, meaning separating it into kind of globules, including separating some pathogens, as well as lysozyme. These are made from the Paneth cells, and they are going to secrete the lysozyme in the intestines here. And then again, lysozyme, we already spoke about up here, is going to be cleaving that peptidoglycan. So in the intestines, we also have another layer or another time that we're releasing lysozyme to specifically attack pathogens, because lysozyme isn't something that's specific to breaking down our food, whereas some of these other things are. This is specific to killing pathogens before they make it further into the body, out of the digestive system and into deeper organs and tissues. Some other body fluid areas are the urinary tract. So in the urinary tract, our urine is actually slightly acidic, so that is meant to inhibit the growth of microbes and also flush them mechanically out of the body. So one of the portals of entry for microbes is through the urinary tract. So moving their way up the urethra and up into the kidneys or the bladder. And our actual urine itself is going to act to flush them out as the urine comes out. But then also, because it's slightly acidic, any of that slight acidity that's left on the urethra is going to assist in preventing the growth of microorganisms and assist in preventing them from getting further into the body. Then we have the female reproductive system. Specifically, cells in the vagina are going to produce glycogen, which is a complex sugar. And our body is going to make this long-chained glycogen strands, and that is because then that's going to be fermented by lactobacilli microbiota. So within the vagina, there's a lot of lactobacillus species. bacteria that are living in there, part of the microbiome. And so they're going to eat this glycogen, this complex sugar with glucose molecules. And then what that does is it produces lactate. And so when that produces lactate, then this is going to lower the pH in that area. So this lactate is acidic. And again, this lower acidity or this lower pH or high acidic environment is going to prevent pathogens from thriving and then prevent pathogens from getting further into the body. Next up, we have our eyes. Our tears contain lysozyme, again, cleaving peptidoglycan. So if we do happen to get pathogens in our eyes, if the wind is blowing and carrying with it a pathogen, and it gets into the eyes, then we have lysozyme in the tears and the moisture in our eyes to break that apart. We also have lactoferrin. Ferrin is related to iron. And lactoferrin is going to sequester iron. So basically what it does is it binds to iron and makes it unavailable for anything to use it. When it makes it unavailable for anything to use it, what that does is it actually ends up starving microbes. And that's because they need iron in order to go through their metabolic processes and sometimes to make toxins and things like that, their virulence proteins. And since they need iron, if we sequester that iron, then it starves the microbes of the iron, and then they're not able to go through their metabolic processes. If they're not able to do that, then they can't continue living, and then they can't produce and infect the body. In the ears, another opening to the environment, we produce cerumen, and cerumen is earwax. So earwax contains fatty acids, and these fatty acids are going to, again, we're going to lower the pH. So within our ears, that earwax is actually helpful. So it's not something that we do want to get rid of. I know some of these things are a little bit counterintuitive with washing the body often and cleaning earwax out of our ears. Of course, we don't want to get to a point where there's such a buildup of earwax that it affects our hearing and causes ear infections or something. But our ears are naturally designed to produce earwax, and that's because they do contain these fatty acids to lower the pH to prevent pathogens, but then also our ears are specially designed to clear that earwax, to continue it moving out away from the body, and that it is essentially sloughed off like the skin cells are. So there shouldn't really be a need to clean that earwax out unless for whatever reason the body's not clearing that earwax out fast enough and then causes a clog or something. So that cerumen is important because it lowers the pH in the ears and prevents pathogens from growing. Then we have the respiratory tract, and in the respiratory tract we have mucus, again, that contains lysozyme, so breaking those things apart. And we also, again, have lactoferrin. Remember, lactoferrin is going to sequester iron. So we have lysozyme that's breaking apart or cleaving peptidoglycan. We have lactoferrin, which is sequestering that iron, that's starving out those microbes. We also have lactoperoxidase in the nasal cavity there. So another enzyme that's going to be breaking down those bacteria, and then other antimicrobial surfactants in the trachea as well as the lungs. So we have surfactant inside of our lungs that makes sure that when we exhale and the air goes out of the lungs and we have a decrease in space within our alveolar sacs, which are the kind of grape-like clusters that fill up with air in our lungs, that when they deflate, essentially, like a balloon would deflate, that they don't stick to each other and stay closed. So we have a surfactant, which is this slippery kind of surface body fluid that allows it to slip back open again very easily when we inhale. So in this surfactant, when it's produced, there are antimicrobial molecules. And again, those are going to hopefully take care of any pathogens that we might accidentally inhale. So for example, as people are around coughing and sneezing and things, and we inhale things, these are different things in the respiratory tract that are meaning to prevent pathogens from being able to continue living and multiplying and then infect the body. So now what we're going to do is talk about some specific chemical defenses called antimicrobial peptides, or AMPs. So AMPs are non-specific, right? So meaning that it's not specific to a particular pathogen, so they're general. They are cell-derived mediators, so coming directly from our cells, so they're an endogenous mediator. And they have broad-spectrum antimicrobial properties. So not only are they not specific, they're not specific to one particular pathogen. They are truly a broad spectrum, like we've spoken about broad spectrum antimicrobials previously, where it's going to affect a wide range of pathogens. These antimicrobial peptides, or AMPs, induce cell damage in different ways. So what they can do is they can damage the cell membranes. So they can go in and they can start to affect the cell membranes and damage those. So we can create holes in the cell and the bacterial cell so that they pop and they can no longer multiply. They also destroy DNA or RNA. So again, if those things are destroyed, then the organism cannot multiply, as well as interfering with cell wall synthesis. So if they can't build a cell wall, they can't make copies of themselves. If they can't make copies of themselves, then they won't be infecting the body. So they may inhibit only certain types of microbes. So some of them, even though they're more broad spectrum, can be something that only affects gram-negative, for example, or only affects gram-positive, for example, or those that have mycolic acid on the surface. Or they can be more broadly effective, so meaning that they can affect bacteria, fungi, protozoa, and viruses, so much more broad. There are many of these on the skin, but then we have some in other areas as well. So for example, we have defensins. These are produced endogenously and exogenously by our microbiome. And these are carried on plasmids that can then also be horizontally transferred. So not only do certain members of our microbiome contain this ability to make these defensins, they can actually transfer this to other ones in our microbiome. So if our microbiome shifts or changes, we start to introduce a new bacterium, then they can transfer it to them and then make these defensins. So here are some AMPs that you should be familiar with. You can see down here, they're the defensins that we were just speaking about, made by epithelial cells, also by macrophages and neutrophils, and we'll talk more about those cells if they're not familiar to you. They're made throughout the body, so where they're made, what body site. And then the pathogens that they inhibit are fungi, bacteria, and then many viruses. So this would be a very broad spectrum type of AMP, antimicrobial peptide. And the way it works is it disrupts the membrane. So anything that has a membrane, if it can disrupt. So many viruses, meaning those that are enveloped, right? Because remember, enveloped viruses are those that have a plasma membrane around them. Back up to the top, we have bacteriocins as the AMP secreted by our resident microbiota. So this is exogenous in our gastrointestinal tract, so our GI tract, meaning the digestive system. And this is going to inhibit bacteria. And again, this is another one that disrupts the membrane. So this one's not quite as broad spectrum, but does the same thing. It disrupts the membrane, but only for bacteria. Then our cathelicidin is made by epithelial cells, macrophages, and some other cell types. This is on the skin, so an AMP that's on the skin. It also inhibits bacteria as well as fungi, and then again, it disrupts the plasma membrane. So lots of these things are disrupting the plasma membrane because, of course, that's where they can first attack. Is this plasma membrane, kind of get through it and burst that cell. The next one is dermacidin. This is made by the sweat glands, or secreted by the sweat glands on the surface of the skin. This is going to inhibit bacteria and fungi. Again, it disrupts the membrane, so it causes the integrity of the membrane to not be very strong, and also it's going to affect the ion channels. So specifically the ability for the bacteria and the fungi to exchange nutrients. And then histatins. These are made or secreted by the salivary glands in the oral cavity, and then this is something that's going to inhibit fungi. So if we accidentally eat some mold on bread that we don't see, this is our first line of defense here with an AMP, antimicrobial peptide, and it's going to disrupt that intracellular function. So if we can break it apart, get into the inside of the cell there with the histatins, then we can hopefully prevent any fungi from moving further into the body. So when we talk about our chemical defenses, we have those AMPs that are produced, the antimicrobial peptides, but we also have some plasma protein mediators. So plasma is the fluid portion of the blood, so the liquidy portion, the part that's not cells. What it does is it contains different types of mediators. So the first mediator is called the acute phase proteins. These are produced in the liver, and they're secreted into the blood in response to inflammatory molecules, which we're going to talk about in just a moment. So these acute phase proteins, again, so what happens is we actually have some sort of response, an inflammatory molecule is made. So there's recognition of a pathogen, let's say, in the body, and then our body is going to start producing inflammatory molecules. These inflammatory molecules are going to find their way via the blood to the liver, and then that's going to make it so that the liver starts producing these acute phase proteins. And then these acute phase proteins are going to be secreted into the blood. We also have complement proteins. So these are circulating constantly in our blood and various other body fluids as precursors that are activated by various factors, so including microbes. So if our body interacts a microbe, then it can start to activate these complement proteins. But these complement proteins are constantly in our blood, moving around our body, so that if we have something that's going to activate it, then they will be activated. We don't have to take the time to produce them, is the point. Then we have cytokines. So our cytokines act as communication signals between cells. So cytokines are ways for one cell that says, hey, I found a bad guy and we need to stop him it's a way that that cell that has identified that microbe, that pathogen, to communicate with another cell, saying, hey, this is what's going on. Those cytokines are going to initiate a response in the other cell. We're going to talk more about each of these as well as we move further throughout the chapter. Again, all of these are part of our non-specific innate immune response. So these are things that just naturally happen. And again, this is non-specific. So when we're talking about an acute phase protein, we're not saying only in response to this particular fungi or only in response to this particular bacteria. We're saying if there's anything in the body that's considered not self, then we end up making these acute phase proteins. If there's anything in the body that is not self, we start to initiate these complement proteins. If there's anything in the body that's not self, we start to make these cytokines to communicate between our cells, saying, hey, there's something that's not self here, we need to do something about it. So when we talk about acute phase proteins, these are, again, antimicrobial mediators. They are produced in the liver, as we said, and then secreted into the blood. And these are some examples of acute phase proteins that you should be familiar with. So, for example, we have C-reactive protein and also serum amyloid A. Both of these are going to coat bacteria, which is called opsonization. So when we talk about C-reactive protein or serum amyloid A, and then other things that we'll talk about moving forward, one of the things that they can do is they actually attach to the surface of the bacteria. So they can attach to receptors on the surface of the bacteria. When they do this, it ends up coating the bacteria, called opsonization. And when this happens, this actually prepares them for ingestion by phagocytes. So these cells that we have in our immune system that are rummaging around, looking out for bad guys, if they interact with the C-reactive protein and the serum amyloid A that are floating around, then it can coat them, and it makes it easier for our phagocytic cells to grab onto them and then ingest them. It also makes it so that they can't adhese or can't attach to wherever it is that they're trying to attach, depending on the pathogen. So remember, that was part of our stages of infection, is that they can attach to something and then get further into the body. But if we coat the bacteria, they can't use their receptors. The next two, ferritin and transferrin, perhaps is familiar to you because of the ferrin piece, which is related to iron. So these are things that are going to bind and sequester iron, and then, as we mentioned before, inhibit the growth of pathogens. We also have fibrinogen. Fibrinogen is involved in the formation of blood clots, and they are very sticky strings that are part of making blood clots, but they also trap bacterial pathogens. So whether it's in that clot themselves or as they're activated outside of the clot area, near the clot, then they're going to act as kind of a sticky net to catch those bacteria. And then mannose-binding lectin. What this is going to do is it's going to activate what's called the complement cascade that we'll talk about. So these acute phase proteins, remember that they are produced in the liver, and that's in response to other inflammatory molecules. So those are going to be in the bloodstream already, go to the liver, and then these are going to be produced. And this is important as we move forward when we talk about mannose-binding lectin. So something has made an inflammatory protein, then the liver is going to make mannose-binding lectin, then mannose-binding lectin is going to activate another type of antimicrobial system called the complement cascade. So the complement cascade, also called the complement system, is where we have different mediators that are going to connect our innate immunity to our adaptive immunity. So we've been speaking about our innate, non-specific immunity so far. And this is stuff, again, that we're born with. We don't develop this over time. It's going to connect this innate response to our adaptive response. And our adaptive response is the part of our immune system that does adapt over time. It does shift and change based on the pathogens that it interacts with. So let's talk about how this works. We have complement activation. All right, so our complement activation is a cascade of protein precursors that are activated, that are triggered by 3 different mechanisms. So our protein precursor activation that's triggered by 3 different mechanisms. So basically there are 3 different ways to cause this complement activation or this cascade that'll happen. The first way that we'll mention is alternative. So the first mechanism that's going to cause this complement activation is called alternative. It's the alternative activation. This is when we have spontaneous activation of the complement protein called C3, or indicated by C3. When this complement protein C3, remember these complement proteins are in our blood plasma floating around all of the time. They also can get into other fluids of the body. So we have these proteins that are part of the complement system floating around in an inactive state. Then when we can have spontaneous activation of C3 in this example. When it activates, it produces C3A and C3B. C3B is going to attach to the microbe, and then it will recruit other complement proteins. So we have C3 floating around as a precursor, then that produces C3A, C3B. C3B is one of these things that attaches to the microbes, and then it tells other cells, hey, this is a bad guy or not self, we need to get rid of it. And we're going to look at this in more detail in the next slide as well. But first, the second one is classical. So this is kind of the first one that they figured out, or the classical method. So C1 is activated by antibody-antigen complex. So in this case, we're looking at the protein C1, the complement protein C1, and it's going to be activated when it comes into contact with an antibody-antigen complex, meaning if we have a pathogen in our body, it has an antigen on it, meaning something that's not self. We will be talking about in Chapter 18 antibodies, but in general, antibodies are something that's produced by our immune system cells, some of them, that are going to attach onto the surface in a receptor key lock sort of way with an antigen. When we have this antibody-antigen complex that's made, what happens is then it activates C1. Then it activates other complement proteins in the cascade. Then the third way is through lectin. So it's activated by the binding of mannose-binding lectin. So remember, we just produced that in acute phase protein from the liver. So when that's released, lectin is activated by that binding of that mannose-binding lectin to a microbial surface carbohydrate. So our mannose-binding lectin is attached to a microbial surface carbohydrate, and then that is going to activate the lectin part of our complement activation, or our complement system. So let's look at the complement system here to make this more clear. So here is our complement system at the bottom. These are our different proteins. So we already mentioned C1, for example. We mentioned C3. And then our mannose-binding lectin is going to go in here at C4. So we said that these things are going to be activated, so they start out in their inactive form, then they're going to be activated, and then we're going to go through this complement cascade. And you can see the different things we just spoke about. So here's our classical pathway. We have our antigen-antibody complex that's going to activate C1. When C1 is activated, then that's going to start to activate all these things down the line here. So that is going to activate C2 and C4, which will activate C3. When C3 is activated, again, we make our C3A and C3B. Then it goes on down the line until we get to this membrane attack complex we'll talk about. The other one that we spoke about, or another one, is the alternative pathway. So again, we're going in at the C3, which is spontaneous activation. That's going to make our C3A and then our C3B again, and then we can continue going down the line here toward the membrane attack complex. And then the other one that we spoke about is the lectin pathway. So our mannose-binding lectin is going to bind to those carbohydrates on a pathogen, and then it's going to activate C4 here, which then is going to go down, activate C3, etc., all the way down to our membrane attack complex. So what we've spoken about so far are these three different pathways to activate our complement system. So then what does the complement system do? Right, so the complement system itself is going to lead to opsonization. And the reason it's going to lead to opsonization is because we create what are called opsonins in this complement system. So we'll talk about that first, and then we'll talk about the other things. So opsonization, again, is the coating of the pathogen by chemical substances. So using these opsonins, these chemical substances that are going to attach to the surface of the pathogen. Once they attach to the surface of the pathogen, allows our phagocytic cells to then recognize that that's not self, and it's able to more easily grab onto that pathogen and then gobble it up, destroy the pathogen. So if we look here, these are our opsonins, are the C1q, C3b, and then C4b. Throughout our complement system, we end up making these things. So this is a simplified version of the complement system, but we can make these opsonins. So our C1q is going to come off of our activation of the C1. Our C3b, we've already spoken about quite a bit, which is an opsonin, which is going to attach to the pathogen. And then our C4b is going to come from the activation of C4. And so these are all different opsonins that are going to go and do opsonization to make it easier for our phagocytic cells to attack and gobble up the pathogen. Our complement system also leads to inflammation, which we'll talk about here in the next slide or moments, as well as chemotaxis. Chemotaxis is movement, taxis is movement in relationship to chemicals, the chemo part. And then also cytolysis. So lysis meaning breaking apart, and cyto, the cell, so killing the cell. So when we talk about the inflammation piece, we actually make our pro-inflammatory complement fragments. So again, we have our complement system at the bottom here, and we mentioned that the complement system, one of its functions, one of the things that it does, is it causes inflammation. And it does this by utilizing these proteins here. So our C3A, which we said can come about via spontaneous activation, but then also down here, when we're kind of moving through and either coming from the lectin pathway or the classical pathway, and we move through and we get to C3b, we can make C3a as well. So we have C3a, and then we have C5a. So C5a down here are both pro-inflammatory complement fragments, meaning that they're going to cause the inflammation process to happen. Which is a good thing because we want to cause inflammation in the body. Inflammation, which we'll talk about later, is helpful for us to clear pathogens and to get pathogens out of the body. So we want those things to be made, and they can be made by the complement system. Another one that we mentioned was chemotaxis, so the movement of cells in response to a chemical. That's, again, the taxis and chemo parts. And our chemotaxis is going to be in relationship to our C5a. So again, down here, not only is it a pro-inflammatory complement fragment, it also attracts white blood cells. So when we have a situation where we have activated our complement system, one of the ways that we can tell our white blood cells about it, our immune system cells, is through this C5a. So we send out the C5a, and then our white blood cells will come to the area where the C5a has been released. That's the chemotaxis. So attracting of the white blood cells to the chemical C5a. Then we get all the way over here. So remember that we are going to initiate this through one of these three pathways. As we just spoke about, moving through these three pathways, we end up making all these kind of intermediate things that we spoke about that are all going to do other things, like opsonization or cause inflammation or cause chemotaxis. But ultimately, the end of the complement system, what it does is it makes the membrane attack complex, or MAC. And this membrane attack complex is, as its name implies, is going to attack the plasma membrane of a pathogen. The way that it does this is it is actually making these proteins here. So what we're doing is we're activating these proteins, not making, but activating these proteins here. And when we activate these proteins, then it's going to assemble these proteins. So we're assembling the C6, 7, 8, and 9. When we assemble them, what we're actually doing is they polymerize, and what they do is they polymerize into pores of gram-negative bacteria. So when we initiate the complement system, if we get to the end, we create the membrane attack complex, and it attacks the membrane of gram-negative bacteria. And it does this because it's going to take these proteins and it's going to put them inside of the membrane, all next to each other, and create a hole. So it kind of creates a tube, kind of like those cell junctions we spoke about in 17.1, where they, the cell junctions that are called gap junctions, where they look like little tiny proteins that are creating these little tiny tubes that go between the cells. Instead of it going between the cells, what we're doing is we're creating these little tiny tubes that are going from the inside of a bacteria cell to the exterior, which is going to deflate, for lack of a better term, but deflate or cause the death of, it'll burst the cell. So the reason that they don't work on gram-positive negative is because the cell wall is too thick with peptidoglycan. So we've got so much peptidoglycan, we're not reaching the membrane to be able to make pores make pores in the membrane that will then burst the cell. So the way that it bursts the cell is because it allows water and ions and other molecules to move into the cell. And then this is going to cause cell lysis. So all of this stuff, if we think about this process, if we make this membrane attack complex and we've got this protein here, and we start to create these pores, and then all of this water starts to rush in. So let's just say these are the pores. I'm creating little holes here. All of this water and these ions and things are going to start to rush in. As it's rushing in, then it's going to end up killing the cell. Okay, so it's going to burst the cell. So ions and other molecules, anything that's in the area, are all going to start to rush in to the cell. And then it'll kill the cell. So that's why it's the membrane attack complex. Just to go back for a moment, remember we have these 3 pathways that go in. We talked about the different things that activating these proteins in the complement system do. And then at the end, we have our membrane attack complex that is useful for gram-negative bacteria. So that wraps up the complement system. So then another type of chemical that is helpful are the cytokines. So cytokines, remember, are communication molecules. These are soluble proteins. They act as communication signals between cells. So these are released to stimulate production of chemical mediators or other cell functions. So for example, proliferation, cell differentiation, inhibition of cell division, and then apoptosis and chemotaxis. So basically, cytokines are a way for our cells to communicate with each other and tell them to do certain things. So they're released to stimulate the production of chemical mediators, like the ones that we've already spoken about. So one cell, if it recognizes there's a bad guy, there's a pathogen, it can release cytokines telling another cell to start making pro-inflammatory fragments, for example, or other types of fragments, or release cytokines itself to go tell other cells to do stuff. But it can tell other cells to proliferate, so to make copies of themselves, and we'll talk about that in Chapter 18. Also cell differentiation, so cytokines can be released to tell a cell to differentiate into a different type of cell that can then attack a bacteria or a complex. Also inhibition of cell division, so if there is some sort of pathogen, then a cell can release those cytokines and tell our body, hey, stop dividing. Don't put your energy into dividing. You should be putting your energy into fighting off some particular pathogen. Also apoptosis. So cytokines can be released from one cell to tell the other cell, hey, you need to kill yourself because you are infected with a virus, for example. And chemotaxis. So again, cytokines are important. For example, when we get a cut and there's bacteria or something in that cut, then chemical mediators can be released, cytokines can be released to tell white blood cells to come quick and get to the area to help get rid of any bacterial pathogens or other pathogen that might have gotten in through that cut, for example. So when we look at cytokines, we have three different functional classifications. The first is autocrine. And our autocrine cytokines is where we have the same cell is going to secrete and receives that cytokine signal. So this would be an example, like we just spoke about, where it's trying to communicate apoptosis. So for example, if this particular cell over here is infected with a virus, it might actually release cytokines. It releases those cytokines that then interact with its own receptors, and then it tells itself to kill itself. Because if it kills itself, then that virus can't take over its machinery and make copies of itself. So that would be an autocrine situation, an autocrine type of cytokine function. The next one is paracrine. So paracrine is when that signal is secreted, the cytokines are secreted, and they go to a nearby cell, so pretty close. So in this image in the middle, we have our secreting cell. That cell is going to secrete those cytokines. Those cytokines are going to interact with the receptor on a nearby cell, and it's going to tell it to do whatever. So it could be apoptosis, it could be, releasing other cytokines, etc., what we had just spoken about. The third kind are endocrine. So our endocrine cytokines are those where we have our signal is secreted into the circulatory system. So it goes into a blood vessel, then it can travel to far, far away cells, so distant cells. So in this last example, here's our cell that's releasing the cytokines. It releases them into the blood vessel, and then they can travel through the blood and end up far, far away to our distant responding cell. And then, again, it's communicating any of those things that we already communicated. So a cell can talk to itself using cytokines. A cell can talk to a neighboring cell using cytokines. Or a cell can talk to a super far away cell via the bloodstream by releasing cytokines into the bloodstream. So then we have different types of cytokines. So now we know what cytokines can do, what they can communicate. We know that they communicate. We know what they can communicate. We know where they can communicate with themselves, close or far away. Now, what are different types of cytokines? So the first type are called interleukins. Interleukins are going to stimulate white blood cells. So this is an example where maybe this would be an endocrine cytokine that is released into the bloodstream. It goes along in the bloodstream, and it's alerting white blood cells to come to an area where there's been an infection, or an invasion, not infection. And that could be done by interleukins. So this is because the term leukocytes are white blood cells. Interleukins are going to be stimulating white blood cells to do their job and perhaps even have some chemotaxis, telling them to come to the area. Another class of cytokines are called the chemokines. Chemokines are going to recruit specific white blood cells to the site of the infection. They can cause tissue damage and cause inflammation. So when chemokines are released, it's going to be telling specific white blood cells to come there. So this is chemical cytokines that are going to be released, and then this would be an example of where we would see chemotaxis. So the movement of those specific white blood cells to come to the area and do what needs doing. They can, however, cause tissue damage, which can be useful if the tissue is infected, right? So if we have cells that are infected with viruses and they release chemokines and it destroys that tissue, well, what it's doing is it's destroying the tissue to try to make sure that those viruses can't multiply and make more and then cause a larger infection. And then also can cause inflammation. So again, inflammation is helpful, and we want that to happen to try to get rid of pathogens. And then the third class of cytokine that we're going to talk about are interferons. And so we have interferons type 1 and interferons type 2 that we'll talk about further. So interferons, our type 1 interferon is produced and released by cells that are infected with a virus. Okay, so in this example here, you can see that we have our cell here that's infected. This is our virus. And what it's going to do is it's going to produce interferons, type 1, and it's going to release those interferons. And the reason it does this is because it's going to stimulate nearby cells to stop production of mRNA, to destroy the mRNA that it's already produced, and to reduce protein synthesis. So when a cell is infected with a virus, it will release this interferon. When it releases the interferon, it's going to go to nearby cells and attach to the receptor. and it's going to tell those uninfected cells to destroy their RNA, for example, and to reduce protein synthesis. So this is because it wants to make the other cells not susceptible. It wants to prevent them from being susceptible to that virus, where that if the cell itself decreases production of proteins, so it's not using as much, it's not using its ribosomes as much, it's not going to be producing as much RNA, therefore decreasing the ribosome functions. Then when the virus gets in, if the virus gets in, it's not going to be hijacking a system that's on full production, that's going through a lot of protein production, and it can just jump right in and then make sure that that cell's making or producing copies of itself. And then it also is going to destroy the mRNA that's already produced, again, to decrease or reduce protein synthesis. It also is going to stimulate immune cells that are involved in viral clearance. So activating the immune cells over here, again, it's released. It's going to go to cells that can then activate immune cells so that we can then attack those virus-infected cells more so. And then it can also tell other cells to undergo apoptosis. So our neighboring infected cells then will be told to undergo apoptosis. So this interferon is released. When it accepts that interferon, or when that reaction or that interaction takes place between the interferon molecule and the receptor, it recognizes that this one's already been infected with the virus, and then it can undergo apoptosis. And then, of course, that last one here is activating immune cells. So type 1 is going to affect protein synthesis and the mRNA. It can stimulate immune cells, and then it can also tell other cells to go through apoptosis. Then we have our type 2, and our type 2 specifically activates immune system cells. So when a cell produces type 2, it's going to not go through and do these other things. This is for type 1, but it does also activate immune system cells. So then the last piece that we're going to talk about in this section is inflammation eliciting mediators. So those mediators that are going to elicit or cause inflammation, which we've already kind of spoken about, we're going to talk about more of these. So the first one is kind of speaking about what we've already spoken about, which are the cytokines. We have these cytokines that can stimulate these acute phase proteins that then act as opsonins, and they activate the complement cascade and then elicit inflammation. So remember all of the stuff that we've already talked about, the acute phase proteins, the cytokines, the opsonins, the complement cascade, all of this stuff can affect inflammation and cause inflammation, as we've already spoken about. But we have some other things. So a mediator that is going to cause or elicit inflammation is histamine. We oftentimes can think of histamine because we'll take antihistamines when we have allergic reactions. So like when people's environmental allergies are acting up, people oftentimes take things that have antihistamines in them. And that's because histamine is something our body is releasing in response to a foreign invader, in response to something that is not self. And it is a pro-inflammatory compound. It's released by basophils and mast cells, and we're going to talk about all those cells in the next section. So we can have cytokines that are produced that are going to bind to our basophils and our mast cells. It's going to tell them to release histamine. When histamine is released, then it's going to cause inflammation in the body. Then we have our leukotrienes. Our leukotrienes are produced from the metabolism of arachidonic acid in the cell membrane of leukocytes and tissue cells. So when arachidonic acid is broken down and is being utilized, we can create these leukotrienes. These leukotrienes are similar to histamine, but they are more potent, so they elicit a stronger reaction, and they are longer lasting. So histamine is kind of a short-term reaction, but it can adjust our bronchioles, so it can mediate bronchoconstriction, which means the constriction or the tightening of our bronchioles, which are the tubes in our respiratory tract in our lungs. So histamine can adjust that, which if you have an allergic reaction, like particularly an environmental allergic reaction, perhaps you're familiar with, kind of the lungs closing and making it more difficult to breathe. And that's because histamine is being released, and that causes bronchoconstriction. And smooth muscle contraction. So other smooth muscle as well, but as that relates to bronchoconstriction is because smooth muscle is surrounding all of the bronchioles or the tubes within the lungs, and so it's constricting them and decreasing our lung capacity, our ability to breathe. However, histamine is more short term. So histamine can last a couple of hours, maybe a couple of days, depending on how often it's released and interacting with these foreign substances. Leukotrienes, however, when they're produced, they're more potent and longer lasting, and they can do things like induce coughing, as well as vomiting and diarrhea. And so obviously more potent, it's not just kind of closing down the respiratory tract to help decrease pathogens from coming in. What this does is actually actively forcing the pathogens out. So via coughing, vomiting, and diarrhea to try to get them out of the body. Next we have prostaglandins, and prostaglandins, again, promote inflammation, so inflammation eliciting mediator, as well as fever, so it helps to increase the temperature in the body. The last one is bradykinin. So bradykinin is going to bind to receptors on cells in the capillary walls. So capillaries are the smallest tubes of our circulatory system or cardiovascular system, so our smallest blood vessels. It's going to cause those capillaries to dilate, meaning making them wider, making them have a greater diameter. It's going to cause them to leak water. So we have more of our blood going through those capillaries. Since we have more blood going through the capillaries and they're opened up more, then it ends up leaking water and also leukocytes, which are white blood cells, into the tissue. And so when we release bradykinin, we're causing inflammation. We're also going to take those little tiny capillaries and make them dilate, leak water, and then white blood cells into the tissues. And this is called edema. And again, edema is helpful to a degree, not extreme, but it's helpful because what it's doing is it's getting more water to the area to kind of mechanically wash away pathogens. But then also it brings white blood cells. So the more fluid, the more plasma, which is the fluid portion of the blood, that gets to an area, the more white blood cells are going to get to that area. Which is important because we want to get white blood cells there so they can take care of any pathogens that may have made their way into that area. So when we talk about inflammation, you're going to see that edema is one of the signs of inflammation. So this could just be in that particular area, not edema to the extreme where, like you may have seen or heard about previously, where somebody's leg or foot or something is incredibly large and that's due to edema. That's an extreme situation that isn't helpful for the body. In small amounts, edema is helpful.